Okay, I want you to imagine this. You're in an emergency room. You're sitting bolt upright on the bed, and you just feel off.
Right, not a sharp pain, just wrong.
Exactly. You're exhausted, maybe a little confused. Your stomach is just churning, and you're trying to explain it to the nurse, but the words feel so vague.
You'd probably say something like, I think it's just anxiety, right? Or maybe I ate something bad.
That's it. And the thing is, the nurse, maybe even the doctor, they might nod along because those symptoms, fatigue, nausea, just feeling unwell, they sound like a million other low-risk things they see every single day.
And that's the danger, that moment, that initial assumption that it's the most common, least threatening thing.
Hmm.
That's where the clock starts ticking on a hidden threat.
Because the textbook picture we all have of a major health crisis, like a stroke or heart attack, it's so dramatic, isn't it?
It is. It's built around these really specific indicators: crushing chest pain, pain shooting down your arm, a sudden facial droop you can't miss.
If you don't have those classic symptoms, the whole sense of urgency just evaporates. They might offer you something for your stomach, talk about stress, and get ready to send you home.
But here's the terrifying part, and it comes straight from the sources you sent over. Those vague symptoms, the exhaustion, the nausea, that mental fog,
Yeah.
that might be your body's unique, sex-specific way of screaming for help.
Wow.
That's right. The research is clear that this kind of presentation, these non-classical symptoms, are often how a stroke manifests in females.
So it's labeled atypical, but it's not actually that rare.
It's not rare at all.
Right.
But the medical system was, for decades, calibrated to the classic male presentation. So it gets misinterpreted.
And what's the consequence of that? A misinterpretation in the ER sounds bad.
It's catastrophic.
Yeah.
Because with a stroke, time is everything. We're talking about clot-busting drugs that have to be given within a really tight window, maybe 4 hours, to prevent permanent brain damage.
And if they're thinking it's just indigestion,
That window closes. Not only are females less likely to get those drugs in time, they're also less likely to be admitted to the intensive care units they need. That delay, just a few hours lost to a wrong guess, can be the difference between a full recovery and a life-altering disability.
this isn't just a few isolated incidents, is it? This feels systemic.
It is. It's a fundamental flaw baked into how medical knowledge was built. For decades, the whole scientific world ran on an unspoken assumption.
The standard male body.
Exactly. And if your biology, your symptoms, your whole disease progression didn't match that male blueprint, you were basically set up to fail.
So that's the core of what we're digging into today. The big question, why? Why are our biologies so different that the exact same illness can look, act, and even respond to drugs in completely different ways?
We're going deep on this one.
Okay, let's unpack it. We are diving into the biological code, the genes, the hormones, even the bacteria in your gut that make diseases so distinct in male and female bodies. This is your guide to understanding why one-size-fits-all medicine is broken and why the future has to be different.
Wait, what? Hold on. Are you seriously telling me that for a huge chunk of modern medicine, they just ignored half the population in their research?
It sounds unbelievable, but yes, it's what we call the crisis of the male default, and it's built on a long, well-documented history of something called the clinical trial exclusion.
And I'm guessing this started with good intentions, or at least what seemed like good intentions at the time.
That was the official line, yeah. For a very long time, I mean, right up until the 1990s in a lot of places, it was just a standard procedure to exclude women of childbearing age from early-stage drug trials.
The main fear being risk to potential pregnancy, right?
Exactly. After huge tragedies like with the drug thalidomide in the 50s and 60s, everyone was understandably hyper-cautious about causing birth defects with experimental drugs.
So that caution, while well-meaning, created this massive scientific blind spot.
A huge one. But it goes beyond just pregnancy risk. There's another reason, a methodological one that really shows the kind of scientific corner cutting that was going on.
Okay, we'll get that.
Researchers basically decided that hormonal fluctuations, the menstrual cycle, made things too complicated.
Too messy.
Too messy, too complex. It would cost more money and take more time to account for those hormonal changes in the research design.
So it was just easier to use a male model, which they saw as this, simple, stable, non-fluctuating baseline.
Precisely. They operated on this totally wrong assumption that male biology was the default human biology, and the cost of that assumption wasn't just theoretical. It led to real, measurable public health disasters.
Okay, give me an example.
There's a landmark report from the U.S. General Accounting Office back in 2001. It was a bombshell.
And what did it find?
It found that of the drugs that have been pulled from the market in the years just before, a huge number of them were withdrawn because they posed greater health risks for women.
So they were approved based on male-only or male-dominated trials.
Exactly. And then when they hit the general population, they started causing serious, sometimes fatal side effects, specifically in women.
Wow.
A classic case is the sleeping pill Zolpidem, Ambien. Years after it was on the market, they did the sex-specific analysis and found that women metabolized it much more slowly.
So they wake up the next morning with higher concentrations of the drug still in their system.
Right. Leading to impaired driving, accidents, all because the original dose was calculated based on a faster male metabolism. The dose was simply too high for the average woman.
That is just a staggering cost for what was essentially scientific convenience. And this didn't just happen in human trials, did it? It went all the way down to the lab rat.
Oh yeah, it goes all the way down to basic animal models. This brings us to another deep-seated bias called the myth of variability.
Let me guess. They thought female mice were also too messy to study.
You got it. For decades, scientists actively avoided using female mice. Their reasoning was that the estrous cycle, their version of the menstrual cycle, would make the data too variable, too unpredictable.
But is that even true? I mean, empirically?
It is not. It's a complete myth. Scientists like Jeffrey Mogul went back and actually analyzed the data from thousands of studies, and what they found was an empirical fact. The variability is, on average, the same in both sexes.
The same. So where did the myth come from?
It came from a selective focus. Researchers focused on the female cycle as this big, inconvenient variable while completely ignoring the sources of variability in males.
Like what? What creates variability in male mice?
Things like dominance hierarchies, fighting in the cages, stress from social status. All of these things cause huge swings in hormones like testosterone and cortisol, which absolutely affect the data. But that was just seen as background noise.
So they picked one source of variability, labeled it a problem, and used it as an excuse to exclude an entire sex from research.
And created a massive blind spot based on a faulty, self-serving assumption. It's a huge deal, and it ties directly into the big health crises we're facing now.
You mean like the fact that U.S. life expectancy has been declining?
Exactly. It's been falling for years. We're lagging behind other wealthy nations. And to figure out why, we need precision. We need to understand the sex-specific factors that are driving these trends. You can't do that if your foundational data is built on only half the population.
It seems so obvious when you lay it out.
Yeah.
So what was the fix? How did institutions start to correct this?
Well, the big institutional solution from the National Institutes of Health, the NIH, was a policy called SABV.
SABV.
Sex is a biological variable. It was a landmark policy. It basically mandated that any researcher wanting NIH funding had to include both male and female cells and animals in their preclinical research.
And they had to account for sex in their human studies too, I assume.
Yes. It was designed to enforce scientific rigor and finally kill that myth of variability. It was a huge step forward.
But I feel like there's a but coming.
There is. This is where things get complicated. According to the source material, in early 2025, a new executive order was issued that targeted certain diversity, equity, and inclusion, or DEI, initiatives in the federal government.
And how did that affect this science-based policy?
The order led federal agencies to scrub or make certain web pages and resources inaccessible. And in the process, all of the NIH web pages about SABV, the guidelines, the scientific frameworks, all of it suddenly vanished.
Wait, they took down the scientific policy resource.
They were rendered inaccessible. And this, as you can imagine, caused huge dismay in the scientific community.
I bet. They're probably arguing that this isn't about politics, it's about biology.
That's precisely the point they're making. The sources we have say scientists see the study of biological sex differences as a fundamental, factual part of improving health for everyone. It's not an ideology.
Right. You can't just substitute a political opinion for a biological reality and expect diseases to suddenly start behaving the same in everybody.
Scientists are now urgently calling for those resources to be restored. They see the SABV policy as essential for good science, for reproducibility, for getting grants. The fear is that if this leads to a failure to fund this kind of research, U.S. healthcare will fall even further behind.
So we've got this historical pattern of exclusion for convenience, and now a very modern threat to the very policy meant to fix it.
It's a perfect storm. We literally hurt ourselves by pretending these differences were too inconvenient to study. But the silver lining is that this challenge has forced researchers to get incredibly creative.
To figure out what's actually driving these differences.
Exactly. We know they're real, but what's the mechanism? Is it our genes or is it our hormones?
It's got to be a mix of both, right? A complex cocktail.
It is. The two main architects are sex chromosomes, so XX versus XY, and sex hormones like estrogen and testosterone. The whole quest of modern biology is to untangle those two.
How do you even do that? They seem so intertwined. If you have XY chromosomes, you generally have tests, which make testosterone. How do you pull those two things apart?
Let's start with the genetic blueprint itself, the X chromosome. This is where we get into what you could call the X chromosome superpower, and its Achilles heel.
Okay, I'm listening.
So females have two X chromosomes. Males have one X and one Y. And the X chromosome is not just some placeholder. It is packed with genetic information. We're talking over 1,000 genes.
And a lot of those are related to the immune system, right?
A huge number. The sources say at least 54 genes on the X chromosome have critical known immune functions.
So right away, there's a problem. If females just expressed both copies of all those genes, they'd make double the amount of protein as males.
Which would be a total disaster for the cell, a genetic overdose. So the body evolved this incredibly elegant solution called X chromosome inactivation, or XCI.
This is where one of the X's gets shut down.
Exactly. Let's use an analogy. Think of your X chromosomes like two powerful graphics cards in a high-end gaming PC. If you ran both at full power all the time, the system would overheat and crash.
So the body's solution is to randomly shut one of them down in every single cell early in development.
Yes. And the key word there is randomly. That randomness is what creates something amazing called cellular mosaicism.
Okay, explain that. Mosaicism.
Think about a calico cat. They're almost always female.
Right, with the orange and black patches.
The genes for coat color are on the X chromosome. Because one X is randomly turned off in each cell, some patches of skin cells end up expressing the gene for orange fur from one X, and other patches express the gene for black fur from the other X. The cat's coat is literally a mosaic of its two different X chromosomes.
So you're saying the same thing happens in humans, but with our immune cells instead of fur color.
Precisely. In a female, some of her T cells are running the program from the maternal X chromosome, and others are running the program from the paternal X. She has two genetically distinct populations of immune cells working together in her body.
Which would make her immune system more robust, more adaptable. It's like having two different antivirus programs running at the same time.
That's the superpower part. It's why females generally have stronger immune responses and are better at fighting off infections. But, and this is a huge but, there's a twist.
The Achilles heel.
The inactivation process isn't perfect. This is the finding that changes everything. It turns out that as many as 20% of the genes on that inactivated X chromosome escape. They resist the shutdown, they refuse to power off. So even on the inactive X, these genes are still being expressed.
So for about a fifth of the genes, females are running both graphics cards at once.
You got it. And this means for some really crucial immune genes, females are expressing a double dose. Think of genes like TLR7, which is a key sensor for viruses. This gives them a massive advantage against infections.
But I'm sensing that trade-off again, an immune system that's always on high alert, running a double dose of certain genes.
It's too powerful. It's much more likely to go rogue and start attacking the body itself. That's the definition of an autoimmune disease.
And the numbers back this up, don't they?
Overwhelmingly. You look at conditions like lupus or Sjogren's syndrome. The female-to-male ratio is as high as 9-to-1. Their genetic blueprint gives them this powerful but very risky immune vigilance.
And the flip side is true for males.
Right. With only one X chromosome, they have no backup. If there's a mutation in a critical immune gene on that one X, they're in trouble.
Like with the CD40L gene mutation.
A perfect example. In males, a defect there causes a severe immunodeficiency, leading to constant life-threatening infections. A female carrier usually have a healthy backup copy on her other X, so she's protected.
Okay, so that's the chromosome side. It's incredibly powerful. But that still leaves the other big question. How do you separate all of that from the effects of hormones? Estrogen, testosterone. They're bathing every cell in the body.
This is where scientists basically had to go into a mad scientist lab mode. They needed to design a special organism just to pull these two threads apart. And that brings us to the four-core genotype mouse model.
The FCG model. This thing is brilliant.
It really is the ultimate tool. So let's break it down. Normally, sex determination is a two-step process. First, you get your chromosomes, XX or XY.
Step 1.
Then, a single gene called SRY, which is normally on the Y chromosome, acts like a switch. If SRY is present, the gonads develop into testes. If it's absent, they become ovaries.
And the testes then produce the testosterone that drives the rest of male development.
Right. So what these FCG researchers did was they hacked the system. It's like going into the source code of a video game. They took the SPRY gene completely off the Y chromosome and put it on a different independent chromosome.
So they could turn on the make testes gene, regardless of whether the mouse had XX or XY chromosomes.
Exactly. They completely uncoupled the chromosomes from the gonads. This let them create 4 different types of mice, 4 core genotypes.
You had your standard female, XX with ovaries, and your standard male, XY with testes.
But then you get the really interesting ones. They could make an XX mouse with testes by adding that little SRY gene.
And they could make an XY mouse with ovaries just by taking the SRY gene away.
They literally swapped the organs independently of the chromosomes. The power of this is just immense.
Because now you can ask incredibly specific questions. You can take the 2 groups with ovaries, the XX and the XY ones, and if you still see a difference between them, what does that tell you?
It tells you the difference is purely chromosomal, has nothing to do with the hormones being produced.
And you could do the opposite. Compare all the mice with testes to all the mice with ovaries, regardless of their chromosomes. If you see a difference there, you know it's a hormonal effect.
It's been absolutely foundational for research on the brain, the heart, the immune system, you name it.
And what the wildest things they discovered with this model is that sometimes the chromosomes and hormones are actually fighting each other.
A biological tug of war?
Totally. They'd find cases where, say, having 2 X chromosomes was pushing inflammation in one direction, but the estrogen from the ovaries was actively pulling it back in the opposite direction.
So the final result, what you'd see on the surface, is only a small difference. You might even dismiss it as insignificant.
But underneath, there are these 2 massive biological forces canceling each other out. Without a model like the FCG, you would never, ever see that complex mechanism at play.
Okay, so now we have the tools. We can separate genes from hormones. Let's talk about the discoveries. What has this new sex-aware science actually revealed?
Let's start with the brain, because the differences there are profound, especially when we look at Alzheimer's disease.
The numbers are just staggering. Alzheimer's hits 2 to 3 times more women than men. And it's not just because women live longer.
No, that's a factor, but it doesn't explain the whole gap. The real story is how sex hormones shape the brain and its vulnerability across the entire lifespan. We have to look at puberty, pregnancy, and menopause as these critical windows.
So it starts early.
It does. During adolescence, hormones are organizing brain circuits differently. For example, males tend to show a bigger increase in the volume of the amygdala, which is tied to emotion.
And females.
Females show a bigger increase in the hippocampus, which is crucial for memory. So right from our teenage years, our brains are being wired differently for how we handle stress and memory.
But the really dramatic shift, the one that's a huge driver for Alzheimer's risk, is menopause.
It's a cliff. Estrogen is incredibly neuroprotective. It's like a constant maintenance crew for the brain. It helps clear out toxic proteins like amyloid beta, it reduces inflammation, and it protects the mitochondria, the little power plants inside our neurons.
And then post-menopause, that entire protective shield just drops.
It drops precipitously, and that loss seems to speed up the epigenetic clock in the brain. It increases stress and vulnerability, basically accelerating the aging process in the very regions that are susceptible to Alzheimer's.
This explains why the timing of things like hormone replacement therapy is so critical, right?
It's everything. The early HRT studies had famously inconsistent results. Sometimes it seemed to help, sometimes it didn't.
And this led to the window of opportunity theory.
Right. The idea is that if you start HRT close to the onset of menopause, when the neurons are still healthy, it can be really beneficial. It reinforces that protective shield. But if you wait 10 or 15 years, when the damage has already started,
It might be too little, too late. The cells are already too impaired to benefit. The timing is everything.
This whole steroid story also gets really complex when you look at neurodevelopmental disorders like autism spectrum disorder, which is diagnosed more in males. This led to the extreme male brain theory.
The idea that high levels of fetal steroids might play a role.
Exactly. Researchers looked at amniotic fluid from pregnancies and found that boys later diagnosed with ASD had elevated levels of a whole bunch of steroids, androgens, estrogens, progestogens, all at once.
Which is biochemically weird.
It's very weird. It's biologically implausible.
Yeah.
In a normal metabolic pathway, if the end product is high, the precursor chemical that comes before it should be low. You shouldn't see everything elevated at the same time.
So the steroids might not be the cause.
They might be more like a canary in the coal mine, a symptom of some other underlying issue with how the fetus or the placenta is regulating steroid production in general. It just shows how much we still have to learn.
Okay, let's switch from the brain to the heart. You mentioned the heart disease switch in diabetes. This one really surprised me.
It's a fantastic example of how crucial nuance is. The general rule is true. Being female is usually protective against cardiovascular disease, even for people with type 1 or type 2 diabetes.
But that's the general rule. The devil is in the details.
The details are the diabetes type and the patient's age. A huge study out of Sweden looking at over 400,000 people found this critical exception that should change how we screen patients.
What was it?
They looked at the risk of having a heart attack. And they found that among women between the ages of 50 and 69, those with type 2 diabetes actually had a lower risk of a heart attack than women of the same age with type 1 diabetes.
Wait, a lower risk? That seems completely backwards.
It is. It totally flips the script. We tend to lump diabetes together as a single risk factor. But this data shows that for a 60-year-old woman, having type 1 diabetes is a much bigger red flag for her heart than having type 2.
So the clinical takeaway is that women with type 1 in that age bracket need way more intensive cardiovascular monitoring.
Far more. More aggressive risk reduction, more vigilance. If we don't make that distinction, we can miss a fatal opportunity for prevention.
So we have chromosomes, we have hormones, but there's a third major player here, right?
The hidden co-pilot. We have to talk about the gut-brain immune axis. Or, more simply, our gut microbiome.
The trillions of bacteria living inside us.
And it turns out sex hormones are like the primary architects of that inner ecosystem. They basically dictate which bacteria get to thrive.
You're kidding. Hormones shape our gut bacteria.
Directly. We have data showing that high testosterone levels, for example, are linked to a gut microbiome that's rich in bacteria like Ruminococcus and Doria.
And high estrogen.
That's linked to completely different bacteria, like Butyrrhizomonas and Slackia. So every time hormones shift in our lives, puberty, pregnancy, menopause, it's causing a major renovation of our internal microbial community.
That link is so powerful it even shows up in our mental states.
It does. There was this fascinating study of archers and shooters. They found that anxiety was linked to specific changes in gut bacteria, and those changes were totally different in men and women.
What did they find?
For one thing, normal, low-anxiety women had much greater microbial diversity than normal men. And when they looked at the anxious women, their gut flora changed, with more bacteria like Faecalibacterium. The low-anxiety women had more of the good stuff, like lactobacilluses.
So our stress levels interact with our gut bacteria in a completely sex-specific way.
It seems so. And this might even be a key to preventing autoimmune diseases.
How so?
In mouse models of type 1 diabetes, it looks like male hormones, androgens, protect against the disease, but only if the right gut microbiome is present.
The microbiome is essential for the protection.
It is. This one experiment was revolutionary. They took the gut microbiome from healthy adult male mice and transferred it into young female mice that were at high risk for developing diabetes.
And what happened?
The transplant didn't just change their gut bacteria, it actually increased the testosterone levels in the female mice.
Wow.
And that, in turn, dramatically reduced their autoimmune response and protected them from the disease. The microbiome itself was acting as a transferable, hormone-modulating protective shield.
The interconnectedness is just mind-blowing. It's not one thing. It's a three-way conversation between our genes, our hormones, and our gut bacteria, all happening constantly.
So we've laid out the problem, the male default. We've seen the biological mechanisms. We've seen the real-world disease differences. The big question now is, so what?
Right. If we know all this, what are doctors and researchers actually doing about it now?
The biggest change is coming from the top down. Regulation is finally catching up. The FDA is in the middle of a major paradigm shift.
What do they do?
In early 2025, they published these 3 huge documents that basically mandate sex-specific data analysis across the entire life cycle of a medical product. It's not a gentle suggestion anymore, it's a requirement.
And this isn't just for drugs, right?
That's the key. It's for medical devices, too. Manufacturers now have to perform sex-stratified analysis and be transparent about it. It's a direct attempt to correct for that historical oversight.
And that regulatory push flows right down to the pharmacy, to precision pharmacology.
Yes. The idea that a drug dose can be one size fits all is officially dead. We know there are profound, predictable sex differences in how our bodies process drugs.
Like in the liver.
The liver is a perfect example. It's the body's main processing plant. There's a family of enzymes called cytochrome P450, and one in particular, CYP3A4, is a workhorse. It metabolizes a huge number of common drugs.
And females have more of it.
And they can have up to twice the amount in their livers compared to males.
So what does that mean for a drug dose?
It means that if a drug is cleared by that enzyme, a standard dose given to a woman might be chewed up and cleared from her system so fast that it never reaches its full therapeutic effect. She might actually need a higher dose.
But for other drugs, the reverse is true.
Exactly. Some drugs are cleared more slowly in women. The same standard dose could lead to accumulation, toxicity, and side effects. We saw this play out in pediatric oncology.
With the leukemia drug 6-mercaptopurine.
Yes. Clinicians found that to get the same effective concentration of the drug in the blood, they had to give significantly higher doses to boys than to girls, knowing that metabolic difference up front is the definition of personalized medicine.
And this all has to be integrated into neurology as well, especially when it comes to those critical windows in a woman's life.
Precision neurology has to be life stage aware. Think about the challenge of treating epilepsy in women of childbearing potential. It is an agonizing clinical dilemma.
Because you have two competing priorities.
Two critical life or death priorities. You have to control the seizures. That is non-negotiable for the mother's health.
But at the same time, many of the anti-seizure medications carry a significant risk of causing birth defects in a developing fetus.
And the risk isn't trivial. The baseline risk of a major malformation is around 2 to 3%. But with a drug like valproic acid, that risk can shoot up to almost 10%.
It's an impossible choice for a patient and her doctor to have to make.
It is. And precision medicine has to deliver safer, equally effective alternatives that are tailored for these specific life stages. We need to understand the mechanisms that make a drug harmful during pregnancy and design our way around them.
So this brings us to the big takeaway, the philosophical mic drop. The goal here isn't just to create two separate playbooks, one for male bodies and one for female bodies.
Well, it's so much bigger than that. It's about moving past that simple binary. The real goal is to understand how our internal sex biology, our chromosomes, our hormones, is in constant conversation with everything else.
With our culture, our diet, our genetics, our age.
And especially our environmental exposures. Scientists have a great term for this. The exposome. It's the sum of every environmental influence on you throughout your entire life.
So this research allows us to finally connect our internal biology to our external world.
It gives us a truly complete picture of individual health that goes way beyond simple categories and helps us optimize health for every single person, however their biology is configured.
Okay, so let's recap the journey. We started with the crisis of the male default model, a system that created dangerous blind spots, like with the dosing of Ambien.
Then we dove into the biology. First, the chromosomes, with that X inactivation superpower that gives a strong immune system, but at the cost of higher autoimmune risk because of those escape genes.
Next were the hormones, acting as lifelong architects of our brains, influencing risk for things like Alzheimer's and even changing how heart disease shows up in diabetes.
And finally, the microbiome, our hidden co-pilot, where hormones shape our gut flora, and that flora in turn influences our immunity and even our mental state.
So, as you're thinking about all this, here's the final thought to leave you with. On average, we know women tend to live longer than men. They have a longer lifespan.
But, and it's a critical but, studies consistently show they often have a poorer health span. They live more years, but more of those later years are spent with disability or poor physical function.
So the question is, if we can really dial in on these sex-specific factors we've been talking about,
if we could leverage estrogen's protective role for mitochondria to fight age-related decline or design a microbiome therapy to boost heart health, specifically in postmenopausal women.
could we do more than just add years to life? Could we finally add life to those years and maximize health span for everyone?
What stands out to you? What's the one biological truth that medicine has been missing for so long that shocks you the most?
Yeah.
Thank you for sharing your sources with us for this deep dive. We'll catch you on the next one.